Assalamu alaikum and a very good morning to everyone present here. My name is Fatma Hissam and I would like to thank everyone for joining us for today's webinar on behalf of the AMU Biochemical Society. Let me start by welcoming our guest for the day, Professor Asad Ullah Khan from the Department of Biotechnology here at Aligarh Muslim University. Welcome, sir, and thank you for finding the time to speak to us about your journey in the world of science. Just a little housekeeping before we get started. If you have any questions during the presentation, please type them into the chat box. And a very good morning to everyone present here. During the question answer session. My name is Fatma Hissam and I would like to thank everyone for joining us for today's webinar. Now, on behalf of the AMU Biochemical Society, let's start this webinar by taking Let me start by welcoming our guest for the day, Professor Asad Ullah Khan from the Department of Biotechnology here at
Assalamu alaikum. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم يا بني إنها إن تك مثقال حبة من خردل فتكن في صخرة أو في السماوات أو في الأرض يأت بها الله إن الله لطيف خبير يا بني أقم الصلاة وأمر بالمعروف وانه عن المنكر واصبر على ما أصابك إن ذلك من عزم الأمور ولا تصعر خدك للناس ولا تمش في الأرض مرها إن الله لا يحب كل مختال فخور واقصد في مشيك واغضض من صوتك إن أنكر الأصوات لصوت الحمير. Translation. Luqman went on saying to his son, "My dear son, in fact, if there be anything to the measure of a grain of rye, and it be hidden in a rock, or in the heavens, or in the earth, Allah will surely bring it forth. Surely Allah is all fine and all aware. My dear son, establish salah and bid the fair and forbid the unfair, and observe patience on what befalls you. Surely this is among the matters of determination. And do not turn your cheek away from people, and do not walk on the earth haughtily. Surely Allah does not like anyone who is proud and arrogant. And be moderate in your walk and lower your voice. Surely the ugliest of voices is the voice of the donkeys."
Thank you, Ariba. Let me start by introducing our society since this is the first webinar that we are conducting. The AMU Biochemical Society was established on the 8th of October 2021 by the students of the Department of Biochemistry from the Faculty of Life Sciences at Aligarh Muslim University. We at ABS aim to help students explore biochemistry as a subject and help them cope with the main challenges during the study by providing access to various academic and extracurricular resources. We hope to inspire engaged students by putting our own spin on the subject by creating a fresh and interactive interface for students from different fields from faculties of sciences and life sciences alike through various fun events, competitions, and opportunities to display their talents. I now hand over the hosting to Ariba Dilshad, who will be taking over from here.
Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning. I, Erbil Shad, another founder of the ABS and a final year student of biochemistry, AMU, take immense pleasure in introducing our esteemed guest for today, who is a microbiologist, biochemist, and a professor at the Interdisciplinary Biotechnology Unit of the Aligarh Muslim University. He received his undergraduate and postgraduate from Aligarh Muslim University in association with the International Center for Genetic Engineering and Biotechnology, New Delhi. Later, along with his lectureship at AMU, he completed his postdoc from Rutgers University. An author of over 240 research articles, he was the top third cited author and had the highest publication during 2008 and 2012 in Aligarh Muslim University, quoted by Skival Solution of Alzheimer's, a worldwide recognized international publishing company. He holds many awards and titles such as the Boys Cast Fellowship of DST, Government of India, the Young Scientist Award, and is the first recipient of the Outstanding Research Award of the Year given by AMU. He is also a recipient of Sri Om Prakash Bhasin Award, and his lab received the Most Active Researcher Award of University in 2009. Not only this, but the Department of Biotechnology of the Government of India awarded him the National Bioscience Award for Career Development, one of the highest Indian science awards. He is also a member of the Scientific Committee of the Indian Academy of Biomedical Sciences. He is also a member of the Task Force on Bioinformatics, Computational and Systems Biology of the Department of Biotechnology. He has also delivered several speeches and talks on different topics. With his nature of philanthropist work, he has also contributed to the welfare of students by conducting various workshops and seminars. The AMU Biochemical Society is extremely honored to have you here with us, sir. Ladies and gentlemen, boys and girls, please join me in welcoming the eminent and the most remarkable Professor Asad Ullah Khan, walking us through his journey in the world of science.
Thank you, Ariba, and the whole Biochemical Society of Biological Sciences of Aligarh Muslim University. Am I audible?
So, yes, sir, you're audible.
Okay, I think there's some connection problem at my side, some power problem. But if you find me that not listening, then please just let me inform somehow through message or so. So I'll again reconnect it because I'm focusing on my talk. So I'm not able to find whether I'm mute or unmute. Okay. Well, so let me start. So thank you very much for a nice introduction. It's a very long introduction, which probably I don't deserve. I thought of when I receive a call from the young girls of Biochemical Society who was established. I mean, it's a good move. And I thought of to give my inspirational talk because for youngsters. And this is the talk I composed right from my PhD, starting from my PhD to date. I'm not covering the more science. A little bit science I'll cover, but the most important here is things that I'll focus how to move towards the career. So without further delay, I'll just start. So this is my first slide, my journey in the world of science. Let me start from here. This is my career when I started my PhD program. I got registered in biochemistry, Aligarh Muslim University back in 1994 with the Professor Masood Ahmed. I think most of you may not know him because he's already superannuated quite a couple of years back. He's a professor of microbiology and molecular biology in the biochemistry department. He was a postdoc in ICGEB when I met him first time. Professor Lal came from Caltech. He was a young scientist in ICGEB, and he asked me that to join my lab, but you have to register somewhere else. So I thought of registering with Professor Masood in biochemistry department, and I was the first Indian fellow who joined ICGEB when ICGEB was in a building of NII and not the separate building as Indian researcher, because ICGEB was meant for only foreign students. But anyway, he was a nice fellow who guided me all the way in PhD. And I started a very unique problem.
Sir, you are
Excuse me,
not
sir.
audible.
Sir, you're not audible.
I found very interesting results, and that's the first time we identify. Am I not audible? Am I not audible?
You were not audible.
Am I audible now?
You were not audible before that.
Now am I audible?
Yes, sir, you're audible.
I don't know why it's so. Okay.
The slide is not visible, right?
Is it visible now?
Yes, sir.
Yes, sir.
Okay. So have you seen all these slides or not? So I will start it from here.
No.
I started my PhD in ICDAB with Dr. Lal, and then move to my research work I was discussing. I did my PhD on T4 RNA splicing, where I found interesting mutants which involved in the RNA splicing, which was published in BBRC later on by Welltaken. And then I moved to my AMU as assistant professor in 97, where I tried to establish my lab, and then I moved for 3 years sabbatical in postdoc, where I work in transcription biology lab in Michael Hamsey in Rutgers. This is a very interesting story we published in Genes and Development, where we proved in SSU72 role, which later on proved to be part of the endonuclease activity, putative and phosphatase activity in transcription biology. And this model was proved through several step of my experiment, where we proved that how SSU72 involved in CPF factor, which involved in cleavage factor of RNA transcript. And this is the first time we identify that how initiation and transcription termination, both in case of yeast system and eukaryotic system, is coupled process. And that result was taken by the Genes and Development in 2003. The paper was well taken by without revision in Genes and Development in 2002. It looks like quite young that time. Michael Hamsey was a Danish advisor, and he was a very even still he's in touch with me, and he's almost 70 years old now, but he's a very active researcher even today. Came back from India, from USA to India. I established my research focusing on antimicrobial resistance. My main focus of my research lab is mechanism. I'm looking at the structure.
Excuse me, sir.
So your audio.
We can't hear you.
On core shell angioedema. Am I audible?
So not from the 11th slide.
I think 11th slide. Am I audible now?
Yes, sir.
I think problem is that the power cut. So net is also problem. I don't know why it's coming this side in the MU. So this is the focus of my focus of research lab, and this is some of the projects I'm working on: metagenomics of pan-India AMR, CRISPR-Cas engineered Nissel strain for probiotics, So focusing that, I'll continue these projects, but I move on Bioscas Fellowship in Italy, one of the stalwarts in AMR, sorry, Professor Raffaele Zerilli, and worked a lot on this lab and learned on AMR techniques, where we published in AAC there. In 3 months' time, we published the OXA 58 story in AAC, one of the top journals. This is the nice Italy, Naples area where this is my lab group. This is my mentor. This is basically some of my graduate and technical students here. Coming back again, I continue my projects focusing on inhibitor design, metallo-beta-lactamases, biofilm pathways, and proteomics and genomics. And recently, we have got very nice projects completed, DFU, chronic infection control using PDT, and metagenomics pan-India is continuing, we are working on. Then again, I moved to Paris on the DBT CREST program, where I worked in Patrice Nordman lab. And this Patrice Nordman lab was one of the authority in AMR. He discovered basically NDM1 with Timothy Walsh in UK together. And there was a story, interesting story with him, that when NDM came, people blame him that why you are giving the name NDM, because name is very much related to Patrice Nordman. They thought they have given the name on his own name, Patrice Nordman. It's not a Nordman, it's in New Delhi metallo-beta-lactamases, but that was the fun part. So he's the one who basically initiated AMR, looking the metallo-beta-lactamases marker, and work a lot on this area. I got a chance to work with him. Again, I'll just give you a glimpse of what research is going in my lab, working on mechanism of AMR, structure in site using the site rectum mutagenesis. Are the slides visible? Because keep updating me.
Yes, sir.
Okay.
Yes, sir, visible, fine.
Okay. So we have generated at least 12 laboratory mutants in my lab.
I guess sir is having a little network issue. It's all right. It's a very common issue. Let us all just bear with him. Sir, sir, you're not audible.
Connected. Please give me time.
It's no problem, sir.
I'm sorry for all inconvenience.
No problem, sir. No problem.
Okay, so I was there in this slide, probably, yeah. Yeah, it's visible. Okay, so we have generated several laboratory mutants. Is it visible now?
Yes, sir. Yes, sir. It's visible.
Okay, we have generated several laboratory mutants, at least a dozen of mutants, and majority of them, they are the function loss mutants. So we try to prove that how the amino acids are responsible to lose the function of the particular metallo-beta-lactamases. So we focus on NDM and CTXM, and the work was taken well in a couple of the journals in the last few years. This is my lab view. Not many students are working, unfortunately, because of COVID, they're not coming regularly. But in another project in CRISPR-Cas inhibition, so we are probably the first, I can claim, where we've published the papers in using CRISPR-Cas inhibitory system designed in our own lab through simple mechanism of the sgRNA and dCas9, and we block several genes like virulence genes, CSG-D genes, and then various pily genes, engineered bacteria formation, right? And that this is the this is the
Excuse me, sir, your voice is cracking.
uh, control. This is the deleted Fimbrian all biofilm is lost here. Am I audible?
Yes, sir.
Okay. So these are some results we publish in 17 to 20, where we have shown that completely loss of Fimbri and several other biofilm causing genes using CRISPR-Cas system. This is my group, at the time when basically she's working on PDT, and he has worked on most of the CRISPR-Cas. One of the girl, Azna, she's not here, but she left Chicago. She's also part, so she has recently joined the Pittsburgh, and many other students are still working in the lab. Now, PDT part of my group, the animal model to generate through PDT, and we completely cure the infection, chronic infection, diabetic foot ulcers through PDT techniques. I'm not going to talk about detailed PDT, but just to give you a glimpse of it. And the work was well taken by core PDT journals in a couple of years. Regarding engineering particles, we are trying to put the antibodies and tagged capping nanoparticles, where we targeting specifically to the target site and using lactin binding and using other tags to the E. coli or to the bacterial system, rather than avoiding it to hitting the other cells of the human system to avoid the toxicity. And that was, again, one of the work in my lab. Cure the caries, see the loss of, this is the rat tooth. We have completely lost the caries, showing the crystal violet binding under the biofilm. So biofilm is completely lost by using these nanocomposite we have used. Besides the nanocomposite, we have also used several other formulation we designed. Then I moved for a couple of weeks to Academia Sinica in Taiwan on INSA fellow in Dr. Su lab. Her lab is basically more on that's a new idea. So we learn a lot from there, and then we try to establish in our lab. She's also providing us a necessary and we're working on the probiotic on CRISPR-Cas system. Coming back on the same aspect, biofilm versus antimicrobial resistance and AMR data. As you know, the reason of why I'm picking the AMR data is because WHO has already announced that it is a priority research area, and down there, by 2050, millions of people will die because of AMR rather than other diseases like cancer and others. And the drug resistance is in 2 out of 3 healthy Indians. This is the study published a couple of years back. I'll just picking some of the quick slides. So this is the example of one of the mathematicians, Indian mathematician based in UK, who died because of infection. No drug was available, and he is a Professor Alexander who was survived because of the availability of the antibiotic, because he has discovered antibiotic by that time, penicillin. And the penicillin was a hope for across the globe, even the battlefield. But now, contrary, having the discovery of such a bullets who can save rather than kill the human, by 2014 there was a meeting, and there was a WHO and CDC organized a meeting and discuss agenda on the AMR, future of AMR, and they found that by 2050, 10 million people will die by AMR than other diseases like cancer, cholera, diabetes, and so on and so forth. So that's one of the pandemic. Quick, we should look into and focus on these problems. 2014, fortunately or unfortunately, my lab discovered NDM4 superbug, and that was a big news covered by several media. But the most important part is not covering the media, but the most important part is that NDM4 was the most severe resistant marker than even NDM1, who were discovered in 2009 by the Timothy Walsh and Patrice Norman group. Quick look into the data, although it is a 2017 published data from my lab, but if you look, the India, Bangladesh are the main reservoir of these dissemination of these resistant markers. So we have to look into India. Then later on identified several laboratories, and we are one of them in the national map, where they identify those who are working in the area of AMR and look into to solve the problem. Aligarh AMU and my lab was number 4th in India out of 360 institutions across the country who are working in this area, and gain a lot to understand about the AMR and different aspects, right from therapeutics and understanding the mechanism in humans, and diagnosis and environmental pollutions. This is the meeting where I've been selected on the beta-lactamases. We develop a very comprehensive database where you can find out the whole markers, country-wise markers, and markers present in the bacteria in order to understand. And that way you can, clinicians and physicians and healthcare workers can get a good idea about the understanding about the whole AMR spread. So that was a meeting in Italy where we discussed about this database, and this is basically Ellen was the stalwart in AMR metagenomics in USA. She will. I keep asking in the between. Am I audible?
Yes, sir.
Yes, sir.
Okay, sure. Okay, thank you. Thank you. Some of the work from my lab, we initiated an ICU of AMU hospital, occurrence of beta-lactam. The MICs goes very high, and that shows that the level of resistance. In another study, we identify, the very rare Serratia lepagi is very rare microorganisms in an ICU, and we genetically identify it through genomics, whole genome sequencing, the kind of a gene which is veiling and carrying these markers from bacteria to bacteria, the IS elements. Again, we characterize some Klebsiella pneumoniae strains from an ICU of AMU JNMC, and we found NDM present. This is the study where we got the 2 patients, and these were basically middle-aged patients of a low-income country, to have some UTI infections. And we got the samples, and unfortunately, both of them could not survive at the end of the treatment. But when we found them to identify the bacteria which are present in uropathogenic bacteria, E. coli, and they were Citrobacter vercumox. And genetically, when we identified, they were found to have this code earlier in other part of the country and globe. But here in Aligarh, we found the first detection, and that was very difficult to treat that patient. Focusing on back to mechanism part, we are looking at the beta-lactamases more oftenly and looking into the structure designing of these beta-lactamases through modeling, and then we mutate this TRP9319 in the endium non-beta-lactam site. Now, these studies, again, I'm not going to detail about these mutagenesis studies, but just to look, you can go through the papers we publish, and you'll find the detail of this, and we found at least 12, almost more than a dozen of mutants, where we identified several amino acids and mobility of the endium. That lab group, again, this is the old pictures. Diwakar has already left, postdoc. He is in, Bakar is in Mayo Clinic, and Abid's in AIMS. He's in Pittsburgh, and she has submitted, and Lama is postdoc, and Seher and Aish projects, where we are trying to look at the pathways to look into using find the target of the drugs to block these activities and to save the infection control. Well, this is my grants, where my project grants, which support me a lot to get all these studies done in last. Those who are helping in one way or the other way, and we jointly publish also together. So with this, I think I'm sorry for all inconvenience. I think this is all because of the power problem, but maybe the next time we'll have a better way to present the next webinar or so, if you invite. With this, I thank you very much, and
thank you so much, sir. That
wish you all the best.
Thank you.
Thank you so much, sir. We know how busy you are, sir, so we're very grateful that you took out the time to come out to our society's first webinar. Thank you very much, sir. Fatma?
Aslam.
Thank you so much, sir. Thank you so much.
So I would like to thank sir for taking out time from his busy schedule and seeing his remarkable work. And he has always been a torchbearer of science, encouraging students like young budding researchers which are trying to get into the field, and he has always encouraged them to follow their passion and go with all the right reasons. So thank you, sir, for taking out time and elucidating your remarkable journey in the field of science. I'm sure it will help a lot of students like budding researchers and a lot of scientists and everyone who is trying to get into carve his own niche in the field of science. So thank you, sir. And I would like to congratulate all the organizers and all the people who have taken this opportunity to join this webinar and have the fortune to hear to sir, and I'm sure that it will help them in their long run. So thank you, sir.
I think some connection problem. Probably the Absar is they have some connection problem.
Yes, sir. So should we go ahead for a question answer session now? So it looks like we already have some questions. So the first question is, what is causing AMR in a country like India?
I think I can have 5 minutes to answer the question. Please.
So the question is, what is causing AMR in a country like India, and what is the way forward in the next decade or so to deal
think
with
voice
this
is not clear.
ever-growing problem? So can you hear me?
I think your voice was not clear. I could not get the question.
Sir, the question is, it's written in the comment box if you could see it there.
Yes.
What is causing AMR in a country like India, and what is the way forward in the next
Your
decade
voice
or
is not
so
clear.
to deal with this? So maybe Ariba could say the question.
No, you are very much audible, I guess. It might be an issue with.
Yes, please go ahead. Yeah.
So there's a question that is, what is causing AMR in a country like India, and what is the way forward in the next decade or so to deal with this ever-growing problem?
Maybe somebody else ask. Yes. I think voice is not clear. I don't know if I'm able to. Am I
Sir,
audible
they
or
are
not?
asking ki what
But
is the
your voice is
reason?
not clear.
So what is the reason? Sir, is mine clear?
Sir, you can read it from the chat box if we are not audible.
Yes, please.
Am I audible, sir? Hello. Can you hear me?
Please go ahead.
Okay, so sir, the question that I asked actually is this: like, what is the main reason that is causing antimicrobial resistance in a country like India, and what do you think should be the way forward in the next decade or so to deal with this particular issue?
Well, I guess Sir's network is acting up, so we can ask Sir's all the questions, and then we'll forward it to you by mail, if that's all right with you all. Hello?
Yeah, sure.
Yeah, so
I think I submitted my question on that response sheet which we filled up on Google Forms. It's the
yeah, we
link was
already
shared.
have many questions. Yeah, we already have many questions
I
from the
think Sir
response
has
forms,
left
and
also.
yeah.
Perhaps there was some technical issue at his end, I guess. So because I
So
was able to
please,
hear all
guys,
of you.
Okay. I was
yeah.
also maybe.
Yeah, so please fill up the feedback form so that we can ensure that you guys get your certificates, and be sure to
Thank you
fill it
for
before
organizing.
leaving. Thank you so much, everybody, for being here. Thank you so much. Also,